Welcome to Evolutionary Biology. Today, we are going to explore the fascinating world of microevolutionary changes. We will discover how tiny, almost invisible shifts in genetics build up over time to create the incredible diversity of life we see around us. By the end of this lecture, you will understand exactly how populations adapt to their environments. To understand what microevolution is, think of a classic chocolate chip cookie recipe. If bakers in different cities tweak the recipe based on local ingredients, the cookies will eventually taste different, even though they are fundamentally the same. This is exactly what happens in nature. Microevolution is defined as the change in allele frequencies that occurs over time within a single population. We're not talking about a dinosaur turning into a bird overnight. We're talking about small, step-by-step tweaks, like mice developing darker fur to blend in with volcanic rocks. Because these changes happen within a single population, we can actually observe them over relatively short human timescales. Now that we understand what microevolution is, let us look at what drives it and why it matters. These genetic shifts are driven by four fundamental forces: genetic mutation, natural and artificial selection, gene flow, and genetic drift. Understanding these forces is absolutely critical for our survival. The most famous real-world example is antibiotic resistance in bacteria. When we use antibiotics, we accidentally create a harsh environment where only the most resistant bacteria survive and reproduce. This is a rapid, dangerous microevolutionary change. Without understanding this process, we would be completely blind to how life constantly adapts, making it impossible to manage diseases, breed resilient crops, or save endangered species. Since we know these changes are happening, a crucial question arises: how do we actually measure them? To measure exactly how much microevolution has caused different populations to diverge, biologists use specific mathematical tools. We need to quantify the genetic differences between subpopulations of the exact same species. By comparing the genetic diversity within a small local group to the overall genetic diversity of the entire species, we can see exactly how isolated and distinct that local group has become. This brings us to a very powerful mathematical index. This mathematical tool is called the fixation index. The formula is straightforward but incredibly powerful. FST represents the fixation index itself. HT stands for the expected heterozygosity, or overall genetic diversity in the total combined population. HS represents the average genetic diversity within the individual isolated subpopulations. When the value of FST is near 0, it means the populations are interbreeding freely and are genetically identical. But as the value increases towards 1, it means the populations are completely isolated and have micro-evolved to become genetically distinct from one another. Let us see this formula in action with a concrete example. Imagine a population of frogs split into 2 separate ponds by a landslide. We are given that the overall genetic diversity of all frogs combined, HT, is 0.50. The average diversity within each individual pond, HS, is 0.40. We want to find the genetic divergence. We substitute these values into our formula. Subtracting 0.40 from 0.50 gives us a numerator of 0.10. Dividing this by the total diversity gives us a final fixation index of 0.20. This number tells us exactly how much they have separated. Now that we have calculated the index, Let us prove what this actually means for the frogs. The numerator we found, 0.10, is crucial. It represents the amount of genetic variation that is due strictly to the differences between the two ponds, rather than within them. By dividing it by the total diversity, we standardize the ratio. An index value of 0.20 mathematically proves that 20% of the total genetic variation is due to the frogs living in different ponds. This proves that microevolutionary changes are actively driving these two populations apart, potentially leading to separate species. We have just seen how to measure divergence between populations. Now, let us zoom in and look at how we track the specific genetic changes happening within a single population over time. Remember, an allele is simply a specific version of a gene, like a variant for brown eyes versus blue eyes. Microevolution is officially occurring when the percentage of individuals carrying a specific allele changes from one generation to the next. To track this, we rely on a fundamental rule of population genetics that links the frequencies of different alleles together. This brings us to the core equation for allele frequencies. The rule is simple. The sum of all allele frequencies for a specific gene in a population must always equal exactly 1. In this formula, P represents the frequency of the dominant allele, and Q represents the frequency of the recessive allele. Because they must add up to 1, if the environment changes and causes the frequency of one allele to increase, the frequency of the other must mathematically decrease. This simple relationship allows us to calculate exact microevolutionary shifts. Let us apply this formula to a population of beetles. We are given that the dominant green allele, P, is initially 0.60, and the recessive brown allele, Q, is 0.40. A severe drought kills the green foliage, making green beetles easy prey for birds. In the next generation, the brown allele frequency rises to 0.55. We need to find the new green allele frequency. We rearrange our fundamental formula to P equals 1 minus Q. Substituting the new Q value, we find the new frequency of the green allele is 0.45. The green allele has significantly decreased due to natural selection. We have found the new frequencies, but let us mathematically prove the exact magnitude of the microevolutionary change. We do this by calculating delta Q, which is the new frequency of the brown allele minus the old frequency. Subtracting 0.40 from 0.55 gives us a positive 0.15. This positive shift mathematically proves that microevolution has occurred rapidly in just a single generation. The beetle population has successfully adapted to the new environmental pressure of the drought and predators, perfectly showcasing the power of microevolutionary change. We have seen how microevolution works mathematically. Now, let's look at how it paints patterns across the globe. When microevolution happens across different geographic areas, it creates interpopulation variations. One fascinating pattern is called a cline. A cline is a gradual, continuous change in a specific trait across a geographical distance. For example, mammals of the same species might have progressively thicker fur as you move from the warm equator toward the freezing poles. There are no sudden breaks or borders here. It is a smooth, continuous gradient of adaptation to changing environmental factors, like temperature. Sometimes, a smooth cline runs into a massive geographical barrier, like a mountain range or a large lake. When the cline bends around this barrier, it creates what biologists call a ring species. Imagine a population spreading in a circle around a vast valley. As they spread, they micro-evolve to adapt to their local spots. Neighboring populations can still mate perfectly fine. However, by the time the two ends of the expanding ring finally meet on the other side, they have accumulated so many changes that they can no longer interbreed. Let us look at this diagram to visualize a ring species clearly. In the center, we have a massive mountain range acting as a geographical barrier. Notice the chain of salamander populations, labeled A through E, forming a ring around the mountain. The arrows between them represent gene flow, meaning neighboring groups can interbreed. You can also see a gradual color shift from blue to red, representing the micro-evolutionary changes along the cline. But look at the bottom, where population A meets population E. There is a red X, indicating reproductive isolation. Even though they are connected by a continuous chain, the ends have evolved so much they act like completely different species. The final pattern we will discuss occurs when populations are completely cut off from one another. When populations become geographically isolated by physical barriers like rivers or canyons, they experience distinct environmental pressures. This causes them to develop into distinct races, also known as ecotypes or subspecies. Unlike the smooth gradient of a cline, races often have very distinct boundaries because of these physical barriers. If these races remain separated for a long enough time, the microevolutionary changes will continue to pile up until they eventually cross the bridge from microevolution to macroevolution, becoming entirely separate species. We have covered a lot of ground today. To summarize, microevolution is the continuous generation-by-generation change in the genetic makeup of a population. We learned that it is always driven by the fundamental forces of mutation, selection, drift, and gene flow. By tracking allele frequencies and using mathematical tools like the fixation index, we can prove and measure these changes with incredible precision. Whether it manifests as a smooth cline, a complex ring species, or distinct local races, understanding these small-scale shifts is the ultimate key to grasping how the magnificent diversity of life on Earth came to be.